Hey guys, today we're going to be talking about proteins. Now, out of the four different macromolecules, I would honestly, and this is just my opinion, say this is the most important macromolecule to know about. We're going to look at proteins a lot more in the next couple chapters, especially at the end of the year when we talk about like genetics and evolution. But proteins are very, very important to biology. Their monomer are amino acids, which we're going to take a look at the structure in a little bit. But they are the monomer for proteins. One of the reasons why proteins are so important is because when you take out all of the water, that means again you have a cell, you take out all the water, you're left with basically just a bunch of proteins. At least 50% of the cell is made up of just proteins. So when you look at somebody, you're looking at a lot of different things, but you're looking at a lot of their proteins. So it's really important that we know what we're really made up of. Now, there are hundreds of thousands of different types of proteins that have been identified. I like to kind of classify and give the analogy that proteins are like the workers of the cell. They do a lot of different functions, and so there are hundreds of thousands out there, or proteins out there that have been identified. I think it's around 80 to 400,000 different proteins that work within our body. There are just a ton of proteins out there, and they're really important. They do a lot of different functions. We're going to talk about why they have certain functions. Here's a quick image of a protein. We can see that proteins, again, are made up of different amino acids. Now, these proteins can be different lengths. They can be very short lengths. They can be very long lengths. But I like the analogy that words in a book are like amino acids in a protein. The sequence of the words in a book determine what the book actually means or what you read in a book. Just like the amino acid sequence can be different, but that amino acid sequence determines what the protein does. One of the words that students can get tripped up on when going over proteins is the word peptide. So again, remember that these amino acids are going to be joined together through dehydration synthesis reactions. That means when we pull a water out between the two amino acids, then they're going to form that strong covalent bond. That bond is given the name of a peptide bond. Don't think there's really anything different for that specific covalent bond. This isn't like a different type of covalent bond. All we do in science is we label our bonds between amino acids as peptide bonds. They're just covalent bonds. We just give them that specific name. If you see a peptide, that means two or more, again, just amino acids joined together. Again, like I said before, that sequence of amino acids determines the shape of the protein. We're going to talk a lot about shape and function in this chapter. I like this image of that protein a little bit more just because you can see there's different colors that represent different types of amino acids. We're going to learn here in a second that there are 20 different amino acids that make up proteins, and their sequence determines the shape of the protein. So again, if we changed one of these amino acids, maybe this color to a red, or maybe this color down here to another purple, like that's going to change the sequence of amino acids. The sequence of amino acids changes the shape of the protein. When you change the shape of a protein, you change the function. So again, the amino acids are like the words in the book. They make up what the protein looks like. Another really important part about the protein is how the protein gets its shape. So there's four different types of structures within a protein. The first is called a primary structure. You can see here primary structure, linear sequence of amino acids. That just means the sequence of amino acids in the protein. So this is like the words of a sentence. How you sequence the words of a sentence determines what the sentence will be. How you sequence the amino acids in a protein determines what the protein is going to be. This would, in my opinion, be the most important because you're laying the groundwork for the protein shape later on. Now, you might ask, how do we know what the amino acid sequence is going to be? We'll save that question for later on. We're going to come back to it later in molecular biology when we see DNA going to RNA going to proteins. But just understand that the primary structure here is how the amino acids are sequenced in a row. All right, so we talked a little bit about amino acids before. Amino acids, remember, are the monomer for proteins. Now, there's 20 different types. This structure is going to show you what the amino acid looks like. I would not memorize this per se, like memorize, what an amino group is, or memorize what a carboxylic acid group is. But I would be able to identify one of these structures going forward. This NH2 functional group, that shows you that that's labeled as an amino group. We have a central carbon. We have a hydrogen group, and we have this carboxylic acid. This carboxylic acid is a carbon double bonded to an oxygen with a hydroxyl group as well. This whole structure right here is known as a carboxylic acid. The reason why they call it an amino acid is because of the amino acid. So this is like the base. This is the base structure for an amino acid. This R group is any type of molecule, any type of functional group that we're going to see attached to the amino acid that determines what the amino acid is actually going to be. So every amino acid will have this top structure. Again, it has the amino group, central carbon, hydrogen, this carboxylic acid. How they vary is because of this R group. I'm going to show you a little diagram here. You can see each one of these amino acids has a different R group. Now, some are going to be nonpolar, some are going to be polar, some are going to be acidic, and some are going to be basic. Do not memorize these. There's a chart that I'll give out to my students that has these, the different qualities, different characteristics of each of the amino acids. But you can see here, each amino acid, like glycine, only has a hydrogen. Alanine only has this CH3 group. Valine has this group right here. They are specific to that type of amino acid. So every valine is going to look exactly like this. And you can see here how the top looks exactly the same between each of them. Now, I understand in this chart, there's one less hydrogen and one more hydrogen here. Ignore that. That's some advanced chemistry stuff. But just understand this part of the amino acid is always going to be the same, and it's the same as this part of the amino acid up here. What changes is this R group, and you can again see every single R group here is different. This is an example of a protein with a quaternary structure. You can see here, this is a red blood cell. We have our hemoglobin molecule. Hemoglobin is made up of 4 different types of polypeptides. You can see here again the red, the purple, the green, and the reddish orangish color that we have here. Again, this is a quaternary structure of a protein. The protein structure is determined by the sequence of amino acids. The amino acid sequence is going to be determined by your DNA. So if we go back far enough, your DNA is going to determine the shape of your proteins. What that means is the reason why you have certain proteins is because of your DNA. Now, again, your protein structure is going to determine the function. So the function of this hemoglobin protein is going to be different than any other protein that you're probably going to have in your body. The structure of it determines the function. I say that 1,000 times in my classroom every year. Structure determines function. If you take one thing away from this lesson, take away the notion that structure determines function. I describe it as, you have a pair of scissors. The structure determines its function. So scissors are really good at cutting paper. Are scissors really good at transporting people around? No, that's the car's job. So a vehicle's structure is really good at transporting people around. It's really bad at cutting paper. The structure of things in life determine the function. The next structure we have is the secondary structure. So again, we went from primary structure, now we're going to secondary structure. How those amino acids bond together and their different characteristics, whether they're nonpolar, polar, acidic, or basic, they're going to start bonding with each other, and they're going to form 2 different things. They're going to form either alpha helixes or beta pleated sheets. You don't have to really memorize how they look per se. You're going to see a lot of these in protein structures that you see online, but just understand that the proteins are now going to start folding on each other, and that how they fold on each other is determined by their primary sequence. So again, the primary sequence dictates how the protein is going to start folding in the secondary sequence. So that's why the primary sequence is so important to a protein, is because it determines how the protein is going to fold or going on. These alpha helixes and beta pleated sheets, they're just going to be hydrogen bonds that form in different ways, and they're going to be formed due to how the amino or acid sequence is in the primary structure. You can see here there's hydrogen bonds forming between the different amino acids in this alpha helix, and it's going to be actually like this corkscrew of a structure. The beta pleated sheets are going to be more like folds of a paper, and they're just going to be forming between the hydrogen bonds. They're going to be forming between, again, the amino acids. You can see here, this is a protein. We have the alpha helixes and the beta pleated sheaths here. Within the protein, you can see those alpha helixes are those corkscrews. The beta pleated sheaths are these big turns. They are signatures that tells you at this point in the protein, we have an alpha helix. In this part of the protein, we have a beta pleated sheath. They are just indicators for where they are in that protein. Our last structure is going to be our two last structures are going to be the tertiary and quaternary structures. Our tertiary structure is when the alpha helixes and beta sheets have already formed, and they're going to start folding on each other, and they're going to create this 3D globular structure. So we went from kind of like a two-dimensional primary structure. We're getting more three-dimensional with this secondary structure. The tertiary structure is really when we see these 3D structures start to form. Again, they start to bond so that these beta pleated sheets and the alpha helixes start to bond with each other due to the primary sequence. This is why, and again, my opinion, this is the most important of the four different structures, is because this is how the primary structure is how the secondary structure bonds. The primary structure is how the tertiary structure bonds. And we're going to see later on that the primary structure is how the quaternary structure forms as well. So again, these alpha helixes and beta pleated sheets are going to start bonding with each other, and they're going to form this this tertiary structure. Within some proteins, not all proteins, we're also going to see this quaternary structure. What the quaternary structure is if you have several different polypeptide strands. So you can see this is one strand of like for a protein. Sometimes the proteins are just going to be one strand of amino acids. Sometimes they're going to be several different polypeptide strands bonded together. Not all the time are you going to have a quaternary structure. Only some of the time. A lot of times you're going to have, it just depends on the protein, whether it's going to end with a tertiary structure or end with a quaternary structure. I'm going to show you this quick video. It's really important. You can see here, this is a protein. It's going to try to find its least energy state. What that means is it's going to bond with each other, and those amino acids are going to you can see here at the end, they're going to bond with each other. They're going to start creating these alpha helixes and the beta pleated sheets. They're going to start forming with each other. And that protein is going to start out maybe looking like this. Again, that's the primary sequence. You can see all the different amino acids there. And by the end, it's going to have this 3D globular structure. The last concept we're going to go over for this chapter is denaturing. What happens when a protein denatures is the temperature of the protein or around the protein goes past a certain point, or the pH changes. So let's say we have a protein at a pH of like 6. Maybe the pH goes to like 2 or 14. That protein's not going to like that environment. So what happens when you change the pH or you heat the protein up too much is that the hydrogen bonds within that protein are going to start breaking. I know that the hydrogen bonds are very weak, but they will start breaking, and they're going to the whole protein is going to start denaturing. What that means is this nice protein that has a structure is going to become unstructured. You can see here how this protein, the normal protein, looks like any type of protein that you've seen. When you denature it, it's going to start breaking those hydrogen bonds, and it's going to really unravel. Once you unravel that protein, you change the structure. When you change the structure, you change the function. And a lot of times when you denature a protein, it loses its function. So that's why, again, when you heat something up too much, or when you change the pH too much, you're going to denature it and you're going to destroy that protein because it can't function properly.